Hi all. Here we are now for our last biomolecule discussion for CHY 107. And believe it or not, our next to last lecture overall. The end is in sight. We are almost there. And so today we're going to get into a discussion of nucleic acids, namely RNA and DNA. And so let's go ahead and dive right in. If I can get my computer to work. There we go. Nucleic acids are polymers which are constructed from what we call nucleotides. A nucleotide contains three main parts. We have a monosaccharide. Remember, saccharides are just sugars. So it's something for a monosaccharide, a phosphate group, and a nitrogenous base. And so three main parts to all different types of nucleotides. And nucleotide is, as the name implies here, nucleic acids, our RNA and our DNA molecules, are made up of long chains of these various nucleotides. The monosaccharides that make up DNA and RNA are slightly different. The sugar that makes up RNA is D-ribose, and the sugar that makes up DNA is 2-deoxy-2-D-ribose. Very, very slight differences. I mean, look at these. Can you see the difference here? Yeah, that's our only difference. Here at carbon 2, whether our hydroxyl group is below or whether we have a hydroxyl group there or we don't. It's a very, very slight difference there, but that ends up making a difference in the sugar, and different sugars make up either DNA or RNA. The sugar we do number from the anomeric carbon going around clockwise, and we'll see why some of this numbering is important as we go on. We do typically number the sugar with a prime in case we are doing something that we also need to number the nitrogenous base, the carbons and the nitrogens here in our base. And so the numbering just differentiates between the two to make sure that if we say we're talking about the 4' carbon of our nucleotide, we know that we're talking about the carbon that's on the sugar and not one of the carbons on our base. And so again, this is just another depiction of a general nucleotide structure with our sugar and our phosphate group and our nitrogenous base attached onto the anomeric carbon of our sugar. DNA and RNA contain mostly the same different types of nitrogenous bases. DNA will have adenine, guanine, cytosine, or thymine. The only difference with RNA is that RNA, instead of containing thymine will contain uracil instead. Other than that, RNA also contains adenine, guanine, and cytosine. But these are the 4 different nitrogenous bases that may possibly make up the various nucleotides in our nucleic acid chains. Just another type of nucleotide to be aware of. It'll come into our discussion for a little bit, but nucleotide triphosphates, very, very common high energy forms of nucleotides. ATP, probably the one that most of you are somewhat familiar with, adenosine triphosphate. And here's a picture of it here. And so these cells in general get their energy from ATP. And they can do so because this phosphate bond here is a high energy bond. So breaking some of these phosphate groups off can release energy that our cells end up using for various things. And so we have our general structure then for our nucleotides. What can then happen is nucleotides will start to bond together, similarly to how in our discussion of nucleic acids, we talked about how amino acids will bond together to begin to form long chains. Nucleotides will also bond together to start to form long chains. And so RNA and DNA are simply linear polynucleotides. They're linear chains of many different nucleotides all bonded together. And we can see that here. What happens is we end up with a covalent bond between the 3' hydroxyl of our sugar So that just means that so remember in our sugar, we had this OH that was hanging down. You can take another look at that here. So in both, sorry, in both DNA and RNA at this 3, we have our OH group hanging down. What that's going to do is it's going to form a bond with the phosphate group coming off the 5' of another. So we can see that yeah, sort of. Let me try to get, there we go. This is better. And so what ends up happening is we have this hydroxyl group off our 3' of our sugar. And then off the 5' of our sugar, we have our phosphate group. And so what can happen then is we end up forming a bond, and we're not going to get into the mechanics of that bond formation, just know that that's what happens, is we end up forming a bond between this hydroxyl group and this phosphate group of another nucleotide. And that can start to form these long chains. And so, and what we can see here is 3 different nucleotides all bonded together. And each of those do have a different nitrogenous base. Adenine here, cytosine here, and guanine here. And we'll see where that comes into play. And those different bases can come into play later on in the lecture. When we're writing a polynucleotide, the convention is that we always start from the 5' end and go to the 3' end. And so in other words, if we were naming this nucleotide, here's our 5' end, and here's our 3' end. So we can see the 5' end is from our unbound phosphate, the 3' end down to our unbound hydroxyl group here. And so we would name this nucleotide, or we would this nucleotide as an ACG nucleotide from the nitrogenous bases, the different nitrogenous bases. That's the only thing different between any of these nucleotides. The reaction that does join the two, it's an alcohol and a phosphate group coming together to form a phosphate ester. Very similar to esterification that we talked about a week or two ago at this point. So very slightly different, but very similar to those basic esterification reactions. These reactions do require energy, and so various NTPs will be used. Various triphosphate groups will be used to get the energy for these reactions. The reaction does always occur between the 5' phosphate and the 3' hydroxyl. That's always the two that'll join in order to grow out our polynucleotide. DNA is composed of 2 of these polynucleotides, twisted around each other in a double helix. This sugar phosphate backbone makes up the outer portion of the helix, and the 2 strands are anti-parallel. So what that means, and we'll see this in the next image, what that means is one of the strands is going to go from 5' to 3', from 5' up to 3' down, and the other is the opposite, will be 3' to 5'. So you can kind of picture it like this, so anti-parallel to each other. Rosalind Franklin, hopefully you've heard of her, based on X-ray data. She was able to find that the DNA was in fact helical. Watson and Crick, who get all of the credit for finding DNA structure, really worked off Rosalind Franklin's work. Rosalind Franklin was a breakthrough, absolutely fantastic scientist, and was the one to discover the helical shape of DNA. That's really not in doubt when we look at scientific history. Watson and Crick were able to take her work a little bit further, and what they really found was that the hydrophilic phosphate and the monosaccharide are on the outside. So they were able to dive into the structure a little bit deeper, but very, very much based on the work of Rosalind Franklin, who unfortunately didn't get her credit until years later. And I wish I remembered the history of this. I should look into it more because it's pretty fascinating. But I don't think that she even got any credit for it while she was alive, which is a shame. Watson and Crick, being men, were the ones that got all the credit. And that's just sort of the way, unfortunately, the way that things happened at that point. There were many, just as a complete aside, many absolutely brilliant female scientists in 18th and 19th centuries who, and even early in the 20th century. Most of this work was done in the 40s and 50s. just didn't get the credit for their work because of the way that society was. And it's a shame because they were some brilliant people who made some absolutely phenomenal contributions to the world of science. Rosalind Franklin being just one example. Anyway, and so here's what we mean. And so here's our sugar phosphate backbone. And so the sugar phosphate backbone, remember that what we're really talking about, I can come back. So here's our sugar phosphate part of the nucleotide. And so this is what we mean when we say the sugar phosphate backbone, is it's this part of the nucleotide, which is really making up the backbone of our helical shape, with the nitrogenous bases then hanging on the inside. And this is what we mean by the antiparallel. So we have this strand coming down here, which is going from 5' to 3'. And then this strand here going from 5' to 3' from the bottom. And then the base pairs then end up in the middle. And they're strung together, they're held together with hydrogen bonding. So again, this one of the things that I love about teaching this course is that we start to see how things start to really come together here at the end. All these things that we talked about earlier on with some dispersion forces, intermolecular forces, hydrogen bonding, and some other topics that we've talked about, we're really seeing how those start to come into play with very large biomolecules like this. And hydrogen bonding is key to the structure of our nucleotides here. DNA would not hold together in the way that it needs to without this hydrogen bonding. Some of what we talk about later on in this lecture with DNA replication and transcription and translation can only happen because of the precise nature of a hydrogen bond. Fascinating stuff. I love talking about this sort of stuff because everything connects. That's what drew me to chemistry, is these connections like this that happen. Anyway, and so we see here this hydrogen bond, for instance, that happens, the hydrogen bonding between adenine and thymine, that's what holds the base pair together. And so we've got these two anti-parallel strands of DNA held together from hydrogen bonding that occurs between the nitrogenous bases that end up in the middle of our sugar phosphate backbone. A nitrogenous base on one strand, as this is just sort of in words what I was just showing, but a nitrogenous base on one strand forms either 2 or 3 hydrogen bonds on the opposite strand in order to form our base pair. The base pairs in DNA are adenine to thymine and guanine to cytosine. There are 2 hydrogen bonds between adenine and thymine, 3 between guanine and cytosine. And the 2 strands of DNA, as I mentioned, are held together by hydrogen bonding as well as by base stacking. And so we get some of this stacking up of the bases, and there are some interactions that happen here as well, which help to hold the whole thing together. And again, just another, we can see that here. So here's our hydrogen bonding holding adenine and thymine together, our hydrogen bonding holding cytosine and guanine together. And we can see here's our backbone, but then our hydrogen bonding holding our base pairs together. And so DNA, working together with very specialized proteins, coils together into chromosomes. Hopefully all terminology that you've heard before, some of this anyway. That the helix of DNA, one of the ways that this happens is the helix of DNA coils around what we call a histone. Those histones then compact together into what we call nucleosomes, which then end up getting packed into the chromosomes of cells. And this is just a blown up picture of that. So we can see our DNA here ends up getting wrapped into this histone structure. So this circular disc here is a histone, gets stacked into this nucleosome. So this nucleosome is just all DNA wound together in this tightly packed coil. Those all end up getting stacked up on top of each other, then into the chromosome, which then ends up residing in the nucleus of cells. And so it's this sort of structure, this sort of packing that allows cells to pack in so much information. Remember that one complete strand of DNA contains all the information you need to make a person. So that is a lot of information. And part of the reason that it all works is because of this tight stacking of the structure as it all comes together. The whole entire complete DNA sequence is known as the genome. Only about 2% of the sequence codes for actual proteins and genes. In 2003, the Human Genome Project was able to map out the sequence of all the genes in the entire human genome. The ability of organisms to reproduce in general requires that an organism is able to replicate. That begins with DNA. DNA replication requires several enzymes to assist, and it requires the unwinding of a section of a DNA parent strand to form two different daughter strands. And we can see that depicted here a little bit. And we're going to see some of this terminology in some different diagrams later on as well. But here's our original helix. And then it begins to unwind, and we have what we call the replication fork here. But then based on that unwinding, we have the parent strand of DNA here in brown, and then a daughter strand of DNA can then form from that parent strand. It all starts with that unzipping of the DNA. Both strands, both parent strands are copied simultaneously from the 5' to the 3' direction, because DNA polymerase, which is one of the enzymes that helps assist this whole process, only functions from 5' to 3'. Replication begins at what we call the replication fork. We have two different strands of DNA that will start to form from this fork. The leading strand is synthesized continuously from 5' to 3'. The lagging strand is synthesized discontinuously in fragments that are then later joined together by another DNA enzyme, DNA ligase. The enzyme helicase unwinds the strand to begin with. And so all these different enzymes, DNA polymerase, remember ACE, that indicates an enzyme. DNA polymerase, DNA ligase, helicase, all these different enzymes at work to help this process. DNA polymerase will insert about 100 nucleotides per second with an error rate of less than 1 per 10,000. Pretty exact process. And then the cells basically proofread. After that, the error rate ends up at less than 1 per billion. There are some still natural mistakes that occur, and those account for some mutations, which we will be talking about. So again, we see sort of a diagram of that here. Here we have our DNA strand, our anti-parallel strand. Here we have our replication fork, so we split to there. We have our leading strand, which is assembled continuously. And then our lagging strand, which are assembled in fragments and then later put together. The main function of DNA, the entire purpose basically of DNA, is that it is a repository of genetic information that is used to build proteins. The central dogma of all of molecular biology, sort of the whole central unifying theme of the entire field of molecular biology, is that DNA is used to make RNA, which is then used to build proteins. And the proteins then go off to do all their various function within our cells. There are 3 types of RNA: ribosomal RNA, messenger RNA, and transfer RNA. And we'll take a look at all of those. Ribosomes, thinking back to your basic biology and your cell structure, remember that ribosomes are complexes of protein and ribosomal RNA, which are located throughout the cytoplasm of a cell. Messenger RNA carries information from the nucleus. So we can sort of see that here, and we're going to get into a little bit more detail in a bit, but here's a nice diagram of what's going on. So here in the nucleus of the cell, we have our DNA. Transcription occurs to form a strand of what we call messenger RNA, That's from the instructions that are in DNA. That messenger RNA then carries that information to a ribosome, where the ribosomal RNA will then actually create the protein. And that process is called translation. And so protein synthesis in general happens in two main steps: transcription and translation. In transcription, in the nucleus of a cell, the segment of one strand of DNA, so one certain segment, a gene, and we'll look at what genes are in a little bit, but one segment, just one tiny little segment of DNA is copied as a complementary single-strand messenger RNA. And so when we say complementary, remember that the structure of DNA, sorry about that. So the structure of DNA, we have our nitrogenous bases, right? And so when we're looking at the structure, that order of nitrogenous bases, we might have a thymine, a guanine, a cytosine, an adenine, another guanine, and so forth. So that's sort of one of the structures of DNA. The complementary strand means that when that messenger RNA is formed, it's going to be the complementary amino acid. So remember that thymine and adenine are paired up, and that guanine and cytosine are paired up. And also remember that in DNA, we use thymine, but in RNA, it's uracil. And so here's our DNA strand. When the messenger RNA strand is formed from that, so the thymine is going to get paired up with an adenine. Guanine would be paired up, would then form a cysteine. The cysteine would form a guanine. The adenine isn't going to form a thymine, but would instead form the uracil. And so this would be the messenger RNA strand, the complementary messenger RNA strand, formed from that little segment of DNA. Then translation. So we have this messenger RNA strand now, this complementary messenger RNA strand. That messenger RNA strand then serves as a template for assembling translational RNA that will contain the necessary amino acids, or the information for the necessary amino acids. So essentially, this strand here then comes to the tRNA to reform. And that has the information needed to form the amino acids, which we'll be talking about how that occurs in just a minute. In transcription, when a certain protein is required, the appropriate gene is expressed. And we'll take a look at what that really means in a minute. And then the template strand is copied as a single strand messenger RNA. The uncopied, or the coding strand, has the same sequence as the messenger RNA. The messenger RNA is then synthesized from 5' to 3' by reading the template in the opposite direction, with uracil, as we mentioned, being substituted for thymine. Every 3 non-overlapping nucleotides, so every 3 nucleotides, is what we call a codon, and specifies for one amino acid, and that's key. So every 3 of these. So here's our template for one certain amino acid. So every 3 nitrogenous bases in DNA codes for a specific amino acid. And so there are 64 possible codons, which constitute the entire genetic code. The genetic code in general is the same for all organisms. These 64 possible codons. Difference is going to be in the orders of some of those. If we get a code of AUG, that codes from a thymine, which means start. We're going to start our protein here. Sorry. There we go. Then we have 3 different stop codons. That means that's the end. So picture like old-fashioned telegrams. Blah, blah, sentence start, blah, 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 stop. Same sort of idea, that the codons will code for the beginning of the sequence and the end of the sequence. The rest of the codons code for various amino acids. Now, if you think about it, this means that amino acids must be coded for by more than one codon because there are only 20. That's all I really need to say there. And so here's where this all sort of ties together. And so here's our RNA strand. And so this RNA strand is what's being brought to the ribosome in order to make the various proteins. And so here's our RNA strand, the first 3, GCU. And so here's our codon list. And so if the first nucleotide is G, that puts us in here. Second nucleotide is C, puts us in here. And so that means that this part, this 3 nucleotide section, this one codon, codes for the amino acid alanine. This second one, ACG, codes for threonine, the amino acid threonine. GAG, this one now codes for glutamine. And so every 3 nucleotides within our RNA strand is a different codon. Each codon codes for a different amino acid. So now take that out even further. Think about that as when we get long and longer DNA strands. We might have a DNA strand that's, say, 150 codons long. Or 150 150 nitrogenous bases. So 150 of these GCUs, all of this in there, 150 of these divided by 3, because every 3 is a codon. That would mean that we would have 50 different codons in there. That means that those 50 different codons code for 50 different amino acids. There's our primary structure of a protein. So think back to when we looked at proteins. Remember, the primary structure of a protein is that sequence of amino acids. That sequence of amino acids comes from the order of codons. Those codons come from this order of nitrogenous bases in the RNA molecule. That RNA molecule was copied from our DNA. And it blows my mind whenever I think about it, the intricacies going on here. So let's go back in that other direction. So we have our cells, have the nucleus in there. The nucleus has chromosomes. Those chromosomes essentially consist of very, very tightly packed, tightly wound DNA. When our body says that it needs a protein, let's say that our body needs more hemoglobin. We need to be able to make more hemoglobin to carry some red blood cells. Our body senses that, so we need more hemoglobin. And so there'll be part of our cells that kick into gear, part of our DNA that says that there's a section of our DNA that's coded for that. Part of that will unzip and copy into the RNA structure for that particular section. That RNA structure is then carried through to different types of RNA and then finally to the ribosome. And then the ribosome is going to read this code on the RNA in order to find the amino acid sequence that it needs to make more hemoglobin proteins. Fascinating, mind-blowing process. It's just really, really, really cool stuff. Biochemistry is cool. Don't tell anyone I said. Don't tell Dr. Woodruff I said that. He's our biochemist. But it's really, really cool stuff. And then translation, sort of talked about a lot of this already, but translation occurs at the ribosomes. Ribosomes consist of two different subunits. There's RNA and there's proteins in there. The larger subunit contains the active site where sort of the action goes on. During protein synthesis, the subunits will lock around the messenger RNA, and the ribosome sort of moves through there, reading one codon at a time. So the ribosome will, so the whole thing kind of, I don't know if I have a diagram of that. Yeah, I sort of do here. And so we get our messenger RNA here, gets locked into this yellow is the ribosome. The messenger RNA gets locked into that. And then the ribosome sort of moves along the pathway here, reading one codon at a time. And so when it reads that codon, it says, oh, it gets to, okay, GGG. And so that's the codon being read. And there's some other stuff going on in there with anticodons that we're not going to get into in that much detail. But that ends up telling us that we need a glycine and so forth. And so that's what ends up happening in our ribosomes, is that messenger RNA gets trapped in there, and the ribosome moves along the messenger RNA and reads the codons to figure out what amino acid needs to be formed. And then actually assembling the amino acid together, that's the job of the transfer RNA, the tRNAs. And those are usually composed of about 75 to 85 different nucleotides. They have this sort of this cloverleaf shape. We can see this here. So here's a tRNA. So we have a tRNA here that is now putting together our amino acid strain. During translation, base pairs end up being formed between the codon of the messenger RNA and the anticodon of the tRNA. We have something called the wobble hypothesis, which allows for non-standard pairings in the third position. We're not going to worry too much about that. Actually, we're not going to worry too much about anything else on this slide. We've already sort of gone over this diagram here. And so this does bring us to mutations. So this whole entire process, things can go wrong. And so a genetic mutation is any sort of permanent change that can happen to one or more nucleotides that can affect the structure of the protein. Because again, come back to here. All it takes is for one of these to be off somehow. Instead of maybe ACG, maybe said this is CCG. So ACG is going to code for, what did we say? ACG will code for threonine. What if it's CCG instead? That's going to code for proline. Totally different amino acid, which could totally throw off our protein structure. And so errors can occur, and those are mutations. Two main types of errors. One of those is substitution, in which one nucleotide simply gets substituted for another. Another type is called frame shift, in which literally the RNA strand kind of shifts down one. And so that ends up shifting the whole thing off. Both of these mutations can affect our amino acid sequence. And so here's a good example here. Here's our DNA strand, our DNA template strand, codes for our messenger RNA. And so this particular code here, we want to get the sequence tyrosine, leucine, alanine, and leucine. Two different types of mutations that can occur. A substitution mutation, so right here, instead of thymine, or instead of guanine, a thymine comes in. So now our messenger RNA codes for an adenine, and so now we get asparagine instead of alanine. Or a frame shift. So in a frame shift, this guanine right here was actually removed, shifted everything off a bit. Now we get valine and tyrosine. These are codons that would code for valine and a codon that codes for tyrosine. And so that can throw off the genetic code. That ends up throwing off the proteins that are actually formed. There are some fail-safes for this. For instance, the fact that the different codons code for the same amino acid, that can minimize the effect of this. For instance, if a mutation produces CCA instead of CCC, So where are we? Yeah, right here. So if we produce CCA instead of CCC, that's still going to create proline. And so there's still some, there's a little bit of that redundancy built in to try to minimize the effect of some of these mutations. Whew, okay, let's talk about some of the effects that these mutations can have, and then we're done. So most mutations are disadvantaged. But there are some that are beneficial. Some women, for instance, have a mutation on their X chromosome, which gives them what we call tetrachromatic vision. A trichromat, most normal vision, people can see a million different hues, different color hues. So picture your difference maybe between as you shift from a yellow to a red and all those hues in between. Our eyes, most normal eyes, can actually differentiate between a million of those different hues. Somebody with a particular mutation that makes them tetrachromatic can actually see 100 million different hues. Most people have 3 cone receptors, which leads to those 1 million hues. Most people have red, green, and blue cone receptors. Colorblind men only have 2, and colorblindness is much more common in men than it is in women, because it is, if I remember right, a recessive X-linked trait. I'm not positive on that. I don't remember my genetics as well as I should, but I'm pretty sure that it's a recessive X-linked, which means that if your mother passes it on to a son, then the son only has that one X and doesn't have another X to compensate. Whereas in women, if they get passed along a mutation where so they only have 2 of these receptors, their other X chromosome might be able to make up for that. And so it's much less common in women. Anyway, tetrachromatic women end up having 4. They have 2 on each X chromosome. That can lead to really cool things like this. So they might see people that are tetrachromatic, instead of seeing this black-eyed Susan like this, they might see it like this, which is really cool. And then just lastly, I'll leave you with this just because I think it's funny. My wife and I go through this all the time. She'll say, "Oh, that's a lovely lavender color." What do you mean, that pink color there? And so this is, don't take this, try to take this just as the joke that it's intended. But how women see colors, oh, look at these different hues. And men, oh, that's red, purple, or orange. That's it. That is all for our discussion of DNA and RNA. I know there's a lot of information packed into there. And so don't feel bad if you took a few breaks through this video. Only one lecture left, and then we are done, believe it or not. And so until then, you stay classy, USM. And thanks for watching.